My research focuses on separations. Separations are applicable to many areas in chemical engineering, and my particular focus initially was in bioseparation. So these are separation processes applied to the manufacture of drugs in the biotech industry, and also biomedical separations. So bioseparations examples include virus clearance during the manufacture of drugs. This is a collaborative project funded by NSF, and the collaboration is with microbiology. And so we look at trying to come up with new methods for removing viruses from the purification of drugs. It turns out this is important for two reasons, not because the manufacturing methods are bad, but because one uses biological systems to make these drugs. For example, you're using cells which have been genetically modified, and most of the cells that are genetically modified today, this is done by using virus vectors, and so the cells can actually produce these virus vectors, which then exist in the product, and FDA requires that you show removal of these vectors, and that's actually a very major problem. That's one. Number two is the use of the cell culture media, which is often from animal-derived compounds. You use bovine serum albumin and so forth. And again, this can lead to possible contamination by viruses that were associated with the animal that survived. Now, there are research efforts to try and get away from animal-derived compounds and so forth, but nevertheless, it's a major problem. So this is a very interesting and exciting project that we're working on. We're trying to validate virus clearance in existing what's called unit operations. These are basically purification steps such as microfiltration, chromatography, etc. The other major area is biomedical separations, and this also comes somewhat from my industrial experience. I've done quite a bit of work looking at designing blood oxygenators, which are used in open heart surgery. And in fact, over 1 million procedures are conducted today worldwide requiring the use of blood oxygenators, so that's a lot. And these devices really are artificial means to keep the patient alive, to oxygenate the blood while the heart is stopped and the surgeon is operating on the heart. And these operations for more severe cases can take up to 6 hours. Another membrane-based project is the use of adsorptive membranes for virus capture. Adsorptive membranes contain specific functional groups on the membrane surface, and these functional groups then bind virus particles and allow other things, other products, other species present to go through. The functional groups could be, say, ion exchange groups, where we try to bind viruses based on their charge, or specific ligands like polypeptides that can be used to attach virus particles. I have already discussed the use of membranes for virus clearance applications. One could use adsorptive membranes here as well. So here you have a feed stream containing the virus particles and product of interest, perhaps a protein molecule. The virus particles bind to the membrane while the protein molecule flows through the pores of the membrane. We also work on the reverse problem, which is to purify and capture virus particles for therapeutic uses. At the moment, I'm collaborating with a group in Germany where we are trying to purify human influenza virus. This virus is very useful and very important in making influenza vaccines, for which the world is there's a great demand. The current manufacturing process is rather slow and tedious, involving use of chicken eggs, and so if we can come up with an alternative cell culture method and then a method to capture the influenza virus, this would be very helpful. So I'm looking at the use of adsorptive membranes for influenza virus purification. We collaborate with microbiology, where we look at the use of adsorptive membranes for capture of densonucleosis virus. This virus is a virus that affects mosquitoes and is of use in trying to develop ways to overcome arthropod-borne diseases that affect humans. So this is another membrane project, and these adsorptive membranes show great potential in virus capture.